Good afternoon and welcome to my talk on developing prokaryotic water quality indicators. My name is Alexander Eiler. I'm a professor at the Department of Biosciences and the Center for Biogeochemistry in the Anthropocene at the University of Oslo. I'm also the founder of eDNA Solutions, an environmental DNA and bioinformatics solution provider. Shortly about the main team behind the work, Domenico Savio from the Karl-Landstein University in Austria, Lorenzo from the Norwegian Institute of Water Research, and Laurent Fontaine from the University of Oslo. So why prokaryotes? You all know very well that bioindicators have become an indispensable part of water quality monitoring and assessment. But it's mainly multicellular eukaryotes that we use for biotic indices. And in contrast, microbes such as bacteria, archaea, and protists are often neglected as bioindicators. And this is despite that they have been recognized for their importance in environmental processes, including biogeochemical, food web, and water quality processes. There's also an impressive and substantial load of literature on microbial communities and how they respond to eutrophication, acidification, and natural anthropogenic organic pollutants through changes in diversity and function. And the current knowledge and data, of course, provides a solid foundation for exploiting microbial taxa and community structure and diversity, as well as functional genes in novel monitoring programs. And of course, these microbial community measures can also be combined into biotic indices, such as alpha and beta diversity measures. And this could potentially improve the resolution of current bioindicators. So throughout my talk, I will argue that by using molecular approaches complemented by advanced bioinformatic analysis, including also machine learning, the assessment of structural and functional diversity of prokaryotes are very promising with respect to acting as bioindicators for both water quality and ecosystem health assessment. So I hope by the end of the talk you are convinced that we should invest time to study microbial community dynamics and that they are a missing link, a very important link for understanding rapid changes in the structure and function of aquatic ecosystems. And that should be addressed in the future by environmental monitoring programs of, for example, freshwater ecosystems. With the exception of tracking microbial taxa, the potential of microbial bioindicators is more or less untapped. And here I show an example of a study we performed along the entire Danube River using both molecular and traditional culturing-dependent methods to assess bathing quality. In the table to the right, you can see how colony-forming units are used to indicate various levels of fecal pollution and thus the quality of the water for bathing purposes. First thing you can observe on the left graph is the good correspondence between the molecular in the y-axis and the traditional indicators on the x-axis. In addition to providing overall assessment, the two molecular indicators that we used also pinpoint to potential sources. The BACHAM is indicative for human sources, while the HF183 is indicative for rumen feces. Considering community-based indicators, already in 2008 we showed the potential of both eukaryotic and prokaryotic community indicators. In collaboration with the Hungarian Danube Research Institute, we performed a study on river epiphyton. Here we assessed both the composition of the epiphyton using traditional microscopic identification and molecular analysis. Back in those days, we used molecular fingerprinting techniques called terminal restriction fragment length polymorphism, short TFLP. So if you look at those dendrograms, you clearly can see overlapping patterns. The top two panels are microscopy-based community dissimilarities, while in the bottom two panels there are community fingerprints from eukaryotic 18S RNA genes and prokaryotic 16S RNA genes. A few years later, in collaboration with US partners led by a PhD student those days, Carolina Fortunato, we made a first attempt to identify prokaryotic indicator species. Here we sampled along the Columbia River and its estuary in the US, and were able to identify prokaryotic taxonomic units indicative for various environmental settings. The method we used here is called indicator species analysis, and it's implemented in the ARC package lab DSV. And this approach was also used to identify key OTUs for seasonal groups within the river, estuary and plum communities, and for spatial groups within the epipelagic community. Here we used analysis of prevalence and the coefficient of variation of taxonomic groups to identify potential indicators. In the left panel, you can see how the various classes behave with regards to prevalence and variation. For example, Gammapodactyria were the most variable classes and also showed high prevalence. On the right, you can see that these highly variable and prevalent taxa showed distinct patterns as revealed by redundancy analysis. For example, Bacilli was positively associated with total phosphorus and Gammapodactyria bacteria with high values of total nitrogen and total organic carbon. And in the light of these results, we suggested that broad microbial indicators can be provided by first identifying taxa with high coefficients of variation and prevalence detected by 16S RNA gene sequencing. And by associating these taxonomic groups with environmental properties, we might also be able to detect prokaryotic biological indicators to be used in setting environmental quality thresholds in aquatic environments. While the scope of this study was too limited to establish generalized relationships between environmental variables and microbial indicators, it still strongly implied that such relationships indeed exist and could be the backbone of powerful bioindicator tools for the future. To overcome the limitations of this Norwegian river study, based only on 24 samples, we used a much larger dataset produced by the joint Danube surveys. Here we could use data from 2 cruises, including over 200 samples collected from the Danube River. And the Danube River is the second largest river in Europe. And here you can see an MDS plot of those 2 cruises. I should mention that there was some differences in the survey designs between the 2 cruises. There was a distinction made between free living and particulate communities in the GDS2, while the GDS3 there was a river cross section used for sampling at each location. And there were also some differences in the molecular assays used. So this might have caused what you see here as parallel patterns, succession patterns observable in this NMDS plot. So thus we divided the data into 3 data sets. We used the particulate associated and the free living samples from the GDS2, as well as the GDS3 samples in separate analyses using the framework that I just proposed based on prevalence variance and redundancy analysis. And this allows us to make allowed us to make some first observation, and these are more general, between environmental variables and potential microbial indicators. Still, I want to emphasize that communities could differ substantially between geological distinct regions, emphasizing the need to use a reference condition approach, such as in the Water Framework Directive, in future biomonitoring with microbial indicators. The last thing I want to talk about now, I want to highlight some general issues with status classification and using indicator systems. Status classification and bioindicators are based on the assumption that ecological associations are monotonic, such as between biodiversity and environmental properties. And monotonic relationships are those relationships that are either positive, negative, or neutral. However, non-monotonicity has been suggested to represent an important driving force in biological systems or ecological systems, because environmental factors are highly variable in both space and time, and organisms do not interact with abiotic and biotic factors in a fixed way. So a common feature of non-monotonic relationships is that they have both increasing and decreasing sectors, as well as different stable states, where the nature of the response can change dramatically when an environmental factor reaches a threshold. A well-known non-monotonic relationship is the example between productivity and biodiversity, as shown in these graphs. In recent work, we show that machine learning methods such as extreme gradient boosting, random forest regressors, and feed-forward neural networks outperform standard statistics when modeling association between biodiversity and environmental properties. You can see fitted surfaces that are far from monotonic. This is actually data, real-world data, showing relationships of organic matter and the bacterial community composition. To interpret the response surfaces, one may begin by looking at the point bordering the diagonal and then follow a line of points further up on the second axis. And you might see a ridge indicates a value next to the yellowish diagonal to be likely a likely threshold from which the shift in bacterial community composition is greater than average. In the same manner, a blue valley indicates a diagonal along which bacterial communities do not shift substantially. And following this interpretation, several thresholds of high variation in bacterial community composition appear. So by using deep learning relationships can be explored with both increasing and decreasing sectors, so non-monotonic relationships. And they will reveal then different stable states. And from an ecological theory point of view, these community patterns can be interpreted as resembling a phenomenon known as hysteresis, where multiple states persist under equal environmental conditions. However, in contrast to alternative stable state theory, which suggests discrete states separate by ecological thresholds, our results point to alternative trajectories in biodiversity in response to an anthropogenic induced environmental change. And of course, this could lead to misinterpretations when using community-based indicators, probably a reason why we should combine various indicators based on both biotic and abiotic variables. So there are some challenges that we still need to overcome before prokaryotic indicators can be implemented in legislation. First of all, the metadata we usually collect in microbial diversity studies is very poor and does not allow the interpretation of responses to multitude of environmental properties. There's also this question of temporal variability. Microbes, particularly pelagic microbes, are very highly seasonal. Of course, we could use benthic biofilms, but instead here we have this spatial variability problem. And as mentioned in my previous slide, there's also this problem of non-monotonicity in natural systems. And there's also no real universal classification system that can be provided to all kinds of questions. And there's probably more, and I'm looking forward to discuss those. With that, I would like to thank you and my collaborators, and I'm open for questions. Thank you.